In this section of the lecture, we're going to be talking about membrane transport. So we talked about the plasma membrane and how the cells have a plasma membrane around them that gives them some sort of barrier. It's like a wall. And substances do have to move inside the cell and be able to move out of the cell. Otherwise, the cell, if it keeps out everything, it's also not going to be able to maintain homeostasis. It needs a constant input of materials and an export of materials. And so that's what this membrane transport allows it to do. There's two main basic methods of transport. There's what's called passive transport, and there's active transport. And basically what this means is whether there's energy required or there's not. So passive means that there's no input of energy required. No input of energy from the cell is required in order to move the substances in or move them out. In active transport, however, you do have to input some energy in order for that to happen. There are a few different types of passive transport. There's what's called simple diffusion, facilitated diffusion, osmosis, and then filtration. The important thing to understand when you're talking about passive transport is something called a concentration gradient. Now, concentration, I'm sure you're familiar with. It's some sort of, it's an amount of how concentrated something is. A gradient is a change over either time or space in different amounts. In the case of passive transport, we're talking about a gradient of concentrations, a high concentration versus a low concentration. So you can think of it kind of like a triangle. So if you have a triangle here, this side of the triangle over here would represent a high concentration, and over here we have the low concentration. So high concentration of some substance moving down to the low concentration. So passive transport processes are always moving from high concentrations to low concentrations. And we either call this moving down the concentration gradient or with the concentration gradient. This is true of all passive processes, moving from a high to a low. So let's look at each of these in turn and figure out what's going on here. So in simple diffusion, so notice here that we have the plasma membrane. This is the phospholipid bilayer made up of all the phospholipids. This phospholipid bilayer is called selectively permeable. And what that means is that it doesn't let everything in, it doesn't let everything out, it's selective in what is able to move in and out. Some things, some substances, the plasma membrane cannot keep out, even if it has no method of keeping these substances out. For example, lipid soluble substances, and this includes things like steroid hormones, which are lipids, cholesterol. Because they are lipid-based, they can move right through the plasma membrane, no problem. There's nothing to hold them back. Because they are attracted to the hydrophobic inside, the insoluble inside of the plasma membrane. They're able to slip right through. Other things can move through as well. We can have things like oxygen can move right through the membrane without any trouble. CO2. These are both very small molecules. Notice there's no charge. They're not ions. They're molecules and compounds that are very small, and they can move in and out of the membrane whenever they want to. There's nothing holding them back. And water, to some extent as well, can move through the membrane, the plasma membrane. So what determines which way they're going to move? Draw our own membrane here, and I'm not going to draw the individual phospholipids. I'm just going to draw a line representing our membrane. Let's talk about oxygen first of all. So let's say this is the outside of the cell, and this is the inside of the cell. This is an oxygen molecule. let's say that we have lots of oxygen outside the cell. There's only a little bit of oxygen inside the cell. Oxygen can move through the membrane, and so which way is the oxygen going to move? Well, oxygen is going to go with its concentration gradient, or down the concentration gradient. We have a large concentration over here, very small concentration down here, and so oxygen wants to move to that lower concentration. This is simple diffusion. So in diffusion, the molecules, whatever they may be, they're simply moving along their concentration gradient or with their concentration gradient. We run into this all the time. So if you're a female, or I mean even a male, if you use cologne, you rely on this idea of simple diffusion in order for that perfume or that cologne that you spray on yourself to diffuse out so that others can smell it. So when you spray the cologne on your body, you have a high concentration of these molecules, these chemical molecules. And then in the air around you, there's a very low concentration. So naturally, these molecules want to move from areas of high concentrations to low concentration. Same thing happens if you were to put a drop of dye into a glass of water. So where you put that drop has a very high concentration of the pigments, and within the water cup as a whole, there's a very low concentration. And so over time, you don't have to do anything. You don't add any energy, and that's an important point. Over time, those dye molecules are going to move from the areas of high concentration to low concentrations until the whole cup has an equal concentration. That's when this is going to stop. That's when simple diffusion stops. As long as there's a concentration gradient, it'll move in or out. But if that concentration gradient goes away, once you equalize, then there's not going to be any net movement in or out anymore. Simple diffusion. The next one is called osmosis, and you've probably heard of osmosis before. It is diffusion, but specifically referring to water, the movement of water. So it's diffusion, simple diffusion, meaning that there's no energy, it's a passive process, no input of energy required, but it's the movement of water molecules as opposed to movement of other things. So I said that the lipid bilayer is soluble to water, so notice that water can move through the lipid bilayer, and it will move along its concentration gradient. So if there's a lot of water outside, a little bit inside, it's going to move inside. Recently, scientists discovered this protein channel as well. So this is a protein channel found in the plasma membrane called an aquaporin, and it allows water to move in and out along its concentration gradient as well. There's a couple ways that the water can move inside and outside the cells. This is important because the concentration of water is going to determine what happens to the cell. And when we're talking about osmosis, though, there's something else that's important, and that's something called a solute. A solute is a dissolved substance, often an ion, found in water. So osmosis is diffusion, meaning the water is going to move along its concentration gradient, but it's another way to think about it is that it's moving to the areas of higher solute concentration. So I'm going to draw here 3 different cells. Well, I did draw 3 different cells. We'll number these cell number 1, cell number 2, and cell number 3. And around these cells, I'm going to draw some solutes, and I'm going to represent them by blue dots. So remember that the blue dots do not represent water, they represent the solutes. So in this first cell, I'm going to put some solutes on the inside of the cell, and then I'm going to put some solutes on the outside of the cell. And the solutes on the inside and the outside are going to be at about equal concentrations. In this cell here, I'm just going to put a few solutes on the inside, and then let's put lots of solutes on the outside. Again, these are solutes such as ions, such as Na positive, Cl negative. The reason this is important is because ions cannot move inside and outside the cell. They are stuck wherever they are. Because they have a charge, they can't move through the plasma membrane by themselves. And then in this cell, let's put a lot of solutes on the inside and just a few solutes on the outside. So the question here is, since we're talking about osmosis, where is the water going to move? What is the net movement of water going to be? So remember, the water in osmosis is moving along its concentration gradient, or it's moving towards areas of higher solute concentration. So let's look at this first picture here. There's about the same number of solutes inside and outside. So that means that there's not going to be any net movement of water. Water will still move inside and outside the cell, but there's going to be no change in that net flow. And so the end result is that cell is going to stay the same size. Let's look at this second cell here. In this cell, we have very few solutes on the inside and a lot of solutes on the outside. In this case, water is going to tend to move outside of the cell. And what is this going to do to the cell? Remember, the solutes can't move, only the water can move. And so over time, this cell is going to shrivel as the water moves out. And it's going to shrivel until the concentration of water to solutes is equal on the inside and outside, or until the number of water molecules per solute, or solute molecules per water, is equal on the inside and the outside. What about this cell down here? We have a lot of solutes inside, very few outside, so the net movement of water is towards the solutes. Another way to think about it is water is trying to dilute the solutes. If there's a lot of solutes, that means there's less water. So the water is rushing in order to equalize the number of solutes per the amount of water that you have. So what's going to happen to this cell? Well, this cell is going to swell. If it swells too much, this cell could eventually burst, and that would destroy the cell. It would die. So you can see how osmosis is very important. And this is important also in the medical field, because depending on the type of IV, for example, that you give someone, depending on the solute concentration of that IV, you can have different effects on the cells in that person's body. Whether those cells shrivel, whether you hydrate those cells and they start to swell, or maybe even get too big, or you give them a solution that is similar in solute concentration to cells in order to just hydrate their body in general. So this is how osmosis works and the effects of osmosis. The last type of passive transport, the second to last because there's also filtration, is what's called facilitated diffusion. Now, facilitated diffusion is very similar to simple diffusion, except that it's called facilitated because that diffusion has to be helped along. So the molecules or the substances, whatever they are, can't move across the membrane all by themselves. It's a passive transport. It's still diffusion because the molecules are moving from a higher concentration to a lower concentration. So notice in this case, we have a lot of these glucose molecules. I guess these are probably representing glucose because of their shape here. Notice they're lipid insoluble. That means that these molecules, and this is true of glucose, glucose is a large molecule. It cannot move across the membrane by itself. It gets rejected. So our cells still need glucose inside the cells. So the way that the glucose gets inside our cells is with these protein carriers. So you have this protein carrier, or a channel, which is like a channel, that picks up the glucose, and then it's going to close and open on the other side, and allows that glucose to move inside the cell. This is facilitated transport. Moving from high concentrations to low concentrations. So there's no energy required here. It's simply moving down its concentration gradient, but it's using a transport in order to do that. There's also solutes that we've already seen, such as Na positive, ions, Cl negative, etc. They also, because they have a charge, they are hydrophilic, meaning that they are attracted to water. And because they're attracted to water, that means that they cannot move across the plasma membrane. Because of this hydrophobic inside here, they cannot move across the plasma membrane. So they're rejected. So the only way to get solutes inside our cells, and we do need solutes inside and outside of our cells, different concentrations, you have to have some sort of channel that allows them to move through. So again, we're going from high concentration to low concentrations, but we're moving down that concentration gradient through diffusion. This is facilitated diffusion. So those are types of passive transport. We have diffusion, osmosis, facilitated transport, and then there's also filtration. And I won't talk much about filtration, but it's simply a type of diffusion that is caused by pressure. So if you have a lot of pressure on the outside, actually let me move back to this slide here. So if we have a high, instead of just having a high concentration, we have a high pressure, the fluid is exerting pressure, that high pressure is going to push those solutes to the inside where there's lower pressure. So pressure can also cause diffusion to happen. We call that filtration. We'll see that when we talk about the kidneys in one of the later chapters. Okay, there's also something called active transport. Active transport is active because it requires energy in order for it to happen. So, we're going to look at how active transport occurs. We'll also look at endocytosis and exocytosis. These are types of vesicular transport, meaning that there are vesicles that are being used, but they are also a type of active transport. So in active transport, the reason energy is required is because in active transport, you are going against the concentration gradient. So diffusion happens because the molecules naturally want to go down their concentration gradient, so you don't have to put any energy. If you want to go back against that concentration gradient, you've got to put in energy. Think of this like rowing a boat down the river. So diffusion or passive transport is if you're headed downriver. You can just sit in your boat, you don't have to paddle, and your boat will naturally move down the river. But if you want to go upriver, now you're going to have to input energy. That's active transport. This happens in a number of places inside our cells. One of the most important is what's called the sodium-potassium pump. In an active transport, you always need some sort of transport protein. Active transport requires energy, and it always has a transport protein. So in the sodium-potassium pump, and this is important in our neurons, we use this. Sodium needs to be moved outside of the cell, potassium needs to be moved inside the cell. But in this case, notice that sodium, and I'm just going to put Na positive, concentration is high, let me fix that, is going to be low on the inside, and the sodium concentration is going to be high on the outside. And so what's happening is our cell is actively, notice the ATP here, is actively taking that sodium, putting it into the transport, and then using the energy of ATP, it pushes that sodium outside of the cell to areas of its high concentration. So you're pushing back against the concentration gradient. At the same time, our potassium is low on the outside, and high on the inside. Potassium is low on the outside and high on the inside. And so using the same energy that we already used here, potassium is going to come into the transport, and then it's going to reopen on the other side and release the potassium inside the cell where it already had a high concentration gradient. So in this case, we're moving from a low concentration to a high concentration for the sodium, and from a low concentration to a high concentration for potassium. That's why we call it active transport. The vesicular transport is also an active transport. In this case, though, we have a whole vesicle that is moving, either inside or outside the cell. So notice this vesicle here, it has a membrane, and it has molecules inside. So if you think about the structures of a cell, where would this vesicle have possibly come from? Give you a second to think about it. If you answered the Golgi apparatus, then you're correct. So remember, the Golgi apparatus packages and transports proteins that have been created inside the cell. So this could be an example of a vesicle that came from the Golgi apparatus. It has these proteins inside. It comes up to the plasma membrane, and it fuses with that plasma membrane and releases all of its contents out into the cell, into extracellular fluid. We call this exocytosis. So think of exo like exit, leaving the cell. Exocytosis is when you're sending large packages of things outside the cell. Here's an example, and this is a picture from your book, but it's showing a vesicle here fusing with the plasma membrane, and all this stuff just being released from the cell. There's also something called endocytosis. Endo meaning in to the cell, so it's opposite of exocytosis. And in endocytosis, is exactly what's happening is we have these materials on the outside of the cell that the cell wants inside the cell, and so it creates a vesicle, an inbudding of its plasma membrane, and when it does that, it's able to ingest those substances that were on the outside. So now we have a vesicle that is formed that is coming from the plasma membrane instead of going to it in endocytosis. And then whatever's inside that vesicle can then be modified by the cell. In this case, we have a lysosome that's coming in that's going to digest that material there. And then we'll release the contents into the cytoplasm where the cell can use it. And then finally, you can get rid of the parts that you don't want and send them back outside the cell in a type of exocytosis. And then the parts of the vesicle can then return to the plasma membrane and be used again. So that is endocytosis. So in both cases, we're moving large substances either inside or outside of the cell. In endocytosis, there's a couple special names for different types of endocytosis. One is called phagocytosis. In phagocytosis, we're bringing in, it's called cell eating. And so, for example, white blood cells are called phagocytes because they ingest things, whether it might be a bacteria or food substances. They ingest things by engulfing them with their plasma membrane and then pulling it into the cell inside a little vesicle and then digesting it. There's also what's called receptor-mediated endocytosis. Notice in this case we have these little receptors, and these receptors are going to be activated by the substances in the extracellular fluid, and when they're activated, it's going to say, oh, what we want is out there, so let's pull it in. And so again, we're making a vesicle and pulling it inside the body. In both cases, we're ingesting things inside the cell, and the only difference here is receptor-mediated endocytosis. I'm going to be a little bit more specific maybe than phagocytosis because these receptors are only going to attach to certain molecules or particles that we want inside the cell. And that is a look at membrane transport and its uses inside the cell.